Now we're going to talk about bacterial gene transfer, which is, this is where we're going to start talking about the weird things bacteria do to improve genetic diversity in their colonies. So vertical gene transfer does not generate diversity. Vertical gene transfer is just the normal way you think of genes being passed on, which is from parents to offspring. For bacteria, this is clonal. So if there's mutations, yes, that can create diversity, but generally speaking, most of the time, vertical gene transfer is clonal. It's not a source of diversity, only in the rare case that there's a mutation. Okay. Now, horizontal gene transfer is what's unique to prokaryotes. Horizontal gene transfer is when bacteria pass genes to their peers, so in the same generation. So that would be like if I just popped out some genes that you wanted and gave them to you. Okay, so if I could just give you some of my genes and you could incorporate those genes into your DNA, that would be horizontal gene transfer. It's not parents to offspring, it's peer-to-peer. And that's something that we can't do, obviously. So just going to remind you about bacterial DNA. So this blue circle, that's the bacterial membrane, that's the cell membrane. So this is a bacterial cell, and then you have this black circle supposed to be the single circular chromosome, and then you've got a whole bunch of these little plasmids, little circular plasmids. Now, the chromosome is where you have all the essential genes for life in bacteria. The plasmids have sort of like bonus genes, and they're self-replicating. They don't require the bacterial chromosome instructions in order to replicate. Thus, plasmids could be said to be endosymbionts of bacteria, or symbionts that live inside the bacteria. They're not living, right, because they're just a bit of genetic material, but they're a lot like viruses. They are just a bit of genetic material, and they have some lifelike properties. So they're self-replicating, and they even encode their own instructions for moving from one bacteria to another. There are 3 horizontal gene transfer methods in bacteria. There's transformation, conjugation, and transduction. Now, transposons and transposable elements are a weird, I separated it from the list. It's a little bit of a weird 4th category. We'll talk about that when I get there. But the main horizontal gene, there's 3 horizontal gene transfer methods: transformation, conjugation, and transduction. Transformation is when when the bacteria picks up naked DNA from the environment and incorporates it into the genome. Why is there, so naked DNA means just DNA floating around outside of a cell. Why is there naked DNA? Well, because bacteria are floating around in their own dead bacteria. So, bacteria live and die within these colonies, and the dead bacteria start to decompose, and all the insides come out, and DNA is just floating around. So, and it just doesn't even have to be the same species of bacteria, because all bacteria are, these bacteria don't live, unless you're growing it in a petri dish, they're not in isolated cultures. Bacteria live in colonies of many, many different species. So bacteria can trade DNA in this way. Now, only a few bacterial species that are competent for part of their life cycle can perform transformation. So not all bacteria can pick up DNA from their environment, but they can pick up DNA from any other bacteria and incorporate it into their genome. Okay. So competent means that the cell wall becomes permeable to large DNA molecules. That's what I mean by competent. So some bacteria, for part of their life cycle, they become competent, meaning their cell wall becomes more permeable to larger molecules like DNA. Because I told you that the cell wall is not a chemical barrier, because it's sort of like scaffolding, but really, really large molecules do have trouble fitting through it. So when the cell wall is more permeable, these certain species of bacteria can pick up naked DNA from their environment. Transformation was discovered in 1928 by a scientist called Griffith. This is Griffith's strep experiment, Streptococcus pneumoniae. So Griffith this is actually also an experiment that indicated that DNA was a genetic code, because in 1928 we knew that chromosomes were responsible for the genetic code, that we inherited some kind of genetic instruction from chromosomes. But chromosomes are equal parts DNA and protein. So at the time, we didn't know if it was the DNA or the protein that was the genetic code. And this experiment that showed us about transformation also contributed to our understanding that DNA is the genetic code. So what Griffith was doing is he was trying to find a vaccine for Streptococcus pneumoniae. And so what he was doing is he was using, he was creating non-virulent strains or dead cultures of strep and inoculating mice with them to see if that would make the mice immune to a virulent, dangerous version of strep. So the way a vaccine works generally, we talked about this a little bit before, but you have to, once your immune system is exposed to a pathogen, it memorizes its chemical qualities and then remembers that for future encounters so that it can nip the infection in the bud. So when you encounter a pathogen for the first time, your body doesn't recognize it as an invader yet. But once you learn it's an invader, once your immune system learns it is an invader, it will remember it forever in the future, or at least for many years, and that gives you immunity. So the way vaccines work is you want to skip the getting sick part, right? So you can inject a person with dead pathogen or with fragments of molecules from the pathogen's surface or with a harmless mutant of the pathogen. And any of those things will, they're dead or they're harmless, they're not going to cause disease, but they still allow your immune system to get to know the pathogen so that you can build immunity. Okay. So what Griffith was doing with Streptococcus pneumoniae is he had two versions for the vaccine. He had some harmful strains that he killed with heat, and he injected those into mice, and the mice did not get sick, but they were immune, and they did not die from, once they were inoculated with the dead, harmful, the once harmful but killed Streptococcus pneumoniae, it made them immune to the live harmful Streptococcus pneumoniae. Then he also was working with mutant Streptococcus pneumoniae that couldn't cause disease. So the mutant Streptococcus pneumoniae, it had a mutation that prevented it from latching on to mouse cells and causing infection. So the mutant Streptococcus pneumoniae was unable to cause disease. He injected the mutant Streptococcus, but the mutants were live. So the live mutants he injected into the mice, they did not get sick, and it did also make them immune to the live harmful Streptococcus pneumoniae. So he had dead harmful, live harmless. What he found is that when he mixed the dead harmful Streptococcus with the live harmless mutants, what he got was harmful Streptococcus again. So when he mixed, so the killed mutants did not kill the mice. The killed harmful Streptococcus, once it was killed, didn't hurt the mice. The live harmless mutants, when they didn't hurt the mice, but when they were mixed together, it did kill the mice. So he figured either the live Streptococcus is bringing, the live harmless Streptococcus could be bringing the dead harmful Streptococcus back to life, or the dead harmful Streptococcus was making the live harmless harmful again, just by being there. Like, it's sort of like the live harmless mutants were absorbing the harmful qualities from the dead ones. What it turns out is it's the second. So if you mix the live harmless mutants of Streptococcus pneumoniae with dead harmful strains, the dead harmful ones are decomposing, their DNA is being exposed, the living harmless ones absorb their DNA, incorporate their genes, their virulence genes, into their genome, and they therefore make themselves harmful. Now, it's not that they know which genes to pick up. There's hundreds, thousands of bacteria in the harmless mutant colony, and they're all picking up different genes from the naked DNA. Well, some of them are becoming harmful strains because they're picking up virulence factors from the dead, harmful bacteria. So yes, it's sort of like they're picking up their essence. What they're doing is they're picking up their genetic code. They're picking up the genetic code for harmfulness, for pathogenicity from the dead bacteria and their surroundings and becoming pathogens because of that. So further experiment, at first Griffith didn't know what was happening, if the dead ones were coming back, the dead harmful ones were coming back to life, or the living harmless ones were becoming deadly. He had to do a bunch of experiments, and in the process of sorting that all out, that's when he discovered that DNA is the genetic code. It's the DNA that was getting taken up into the living harmless mutants, making them pathogenic. Right? So that is transformation, and that's something that just prokaryotes can do. So some species of bacteria, like Streptococcus, can pick up the naked DNA from dead bacteria in their environment, incorporate it into their own genome, and therefore pick up qualities from those dead bacteria. All right, conjugation. Many bacteria can do this, but not all bacteria can do this again. So again, some bacteria can do transformation, not all. Some bacteria can perform conjugation, but not all. Now, this can occur between any genera of bacteria that have the capability. It does not have to be the same species. So this is what sex pili are used for. So when we talked about bacterial anatomy, we talked about these appendages, and we talked about sex pili. So here's where sex pili come in. It's for conjugation. F+ plasmids. So there's these plasmids called F+ plasmids, and they code for the conjugation enzymes. They code for the sex pili. They code for the ability to move the plasmid around. So what happens is there's an F+ plasmid, and the bacteria that has the F+ plasmid will create a sex pili. The sex pili will connect with another bacteria, and the F+ plasmid will move from the original bacteria, the donor bacteria, to a recipient bacteria. Now, only the F+ plasmid is transferred. So there's this plasmid, it codes for conjugation enzymes. It might have a few other genes on it too that are unrelated to conjugation. So anything that happens, so the F+ plasmid codes for conjugation, it codes for the sex pili and other related enzymes, and it might have other genes in the plasmid as well that are, for other things. But it's only that plasmid that can be transferred. Now, in conjugation, when we talk about conjugation for humans, we mean sex. Now, it's not sex for bacteria, but there are elements akin to sex. So, for example, you need compatible mating pairs. You need an F+ bacteria and an F- bacteria. The F+ bacteria is the bacteria that has the F plasmid. The F- bacteria is the one that doesn't have it yet, but is going to receive it. You also need physical contact between cells with the sex pili. Okay? But it's not true sex. Okay? So note, in mycobacteria, remember leprosy and tuberculosis? In mycobacteria, the whole chromosome can be transferred, not just the plasmids. And there's also substantial blending of the two parent genomes in mycobacteria. So in mycobacteria, they're really weird, and it's a lot more like true sex in mycobacteria. So it's a lot more reminiscent of sexual reproduction that you see in eukaryotes. But in most cases, conjugation is not sex. So even though the typical method of conjugation requires 2 bacterial cells, it's not sex because in sexual reproduction, you're blending 2 full sets of genetic information. A full set of genes from a mother and a full set of genes from a father. And the 2 get blended. Well, that's not true in bacterial conjugation. In bacterial conjugation, it's not the full genome that's getting blended. It's just in typical conjugation, in typical bacterial conjugation, it's just the F+ plasmid moving from one bacteria to a recipient, right? There's not a blending, there's not an equal blending of genetics. So that's why it's not true sex. Okay. So there are so I told you mycobacteria are a little weird because the full chromosome can be transferred and there's substantial blending of 2 parent genomes. So mycobacteria have something a lot more like eukaryotic sex, but there's other weird examples. So you might remember this picture down here. These are the roots of legumes, which are like beans and related species. And remember, they have these little nodules in which they house the anaerobic bacteria that can fix nitrogen from the atmosphere into a form that's usable for the plant, right? So even though conjugation usually happens between bacteria, Rhizobacteria, which take nitrogen gas and make it into a usable form, actually can insert plasmid genes into the cells of their host plant, and that is what causes the host plant to produce important things that the bacteria needs to engage in this symbiosis. That is what causes the plant to make these nodules. The plant doesn't have the genes to make this nodule. The rhizobacteria engage in conjugation with a eukaryotic cell, with the plant cell, to put the genes for this symbiosis into the plant cells so that the plant can make these nodules in which the bacteria live. Okay, so conjugation usually happens between two different bacterial cells, but in rare cases it can happen across kingdoms, or excuse me, across domains, actually, across from the bacterial domain to the eukaryotic domain. And here's one example. Transduction. This is the third of the three. This is when bacterial genes are carried to another bacteria through a virus. Genes in this way can be carried across bacterial species, and all species of bacteria are vulnerable to this because it's not controlled by the bacteria, it's controlled by a virus. So you have to, this will make a lot more sense when we cover viral, the viral life cycle. So there are viruses called bacteriophages. Bacteriophages infect bacteria. Phage means virus. A bacteriophage is a virus that infects a bacteria. Okay. And what bacteriophages do, and I will, I'll put up this YouTube link for you so that you can see this. What bacteriophages do is they inject their viral DNA into the bacteria. The bacteria starts expressing viral genes. So the bacteria starts, we talked about transcription translation. The bacteria starts transcribing and translating viral genes. First viral genes will create enzymes that cut up the bacteria's genome. The next viral genes will be for the viral capsid. And then also the bacteria is going to replicate the viral DNA. So the bacteria is creating viral proteins and enzymes. The bacteria is cutting up it's creating viral enzymes that's going to cut up its own bacterial genome, and it's copying the viral DNA. The bacteria is not actually doing this. The virus is hijacking. That's why we call, it's hijacking the bacteria, hijacking the DNA replication equipment, hijacking the transcription translation equipment. Okay. And so the virus basically just takes over the gene expression stuff from the bacterial cell and uses it all for itself and stops the bacteria being able to use it for the bacteria. So now, once all the viral DNA is copied, so there's lots and lots of viral DNA now, there's lots and lots of viral proteins, capsid proteins, they all self-assemble. The viral proteins, the viral capsid proteins, the capsid is that protein case that goes around the DNA, they spontaneously come together. They're just like their chemistry and their shapes, they just automatically lock together on their own around the viral genome, and then they burst out of the cell. The thing is that there's no one directing this process. It's just spontaneous. The pieces just come together around the DNA, and sometimes mistakes happen. Actually, a lot mistakes happen. So I told you that the virus has cut up the bacterial DNA. Well, sometimes the viral capsid proteins come together around bacterial DNA. So now you have viruses leaving, the viral capsid proteins leaving that are carrying not viral DNA but bacterial DNA. When that gets inserted into the next bacteria, it includes bacterial DNA that gets inserted into the new bacteria. Okay? So sometimes viruses take bacterial DNA with them and insert it into the next bacteria that it infects. So you can watch a video of that. We'll talk about that a lot more in the viral chapter, which is also for this week. All right, transposons. Now, transposons you find in both the bacterial world and in the animal and plant world, in the eukaryotic world. So transposons are small segments of DNA. They encode genes that move them from one part of the DNA to another. So in the transposon code are instructions for cutting out the transposon, moving it, and inserting it elsewhere in the chromosome. So you can find these in eukaryotes and in prokaryotes. They are as rare as mutations, which means that they exist, but they're still rare. Okay. Now, these can be beneficial in bacteria because they can also include certain beneficial genes which can be moved to plasmids, maybe even to an F factor, allowing the gene to be spread. So what I mean is, let's say there's a transposon in the chromosome of a bacteria, and it includes a gene for, I don't know, some metabolic protein to make a vitamin or a digestive enzyme. Now, let's just say this transposon cuts around that gene when it moves itself from the chromosome to a plasmid. So we talked about how chromosomes don't get moved through conjugation, but plasmids do get moved by conjugation. So now a gene that was in the essential bacterial chromosome for this species of bacteria is now possible to move to a new bacteria through the plasmid. Now, so there's this title called transposable elements. Transposable elements include transposons, viruses, and plasmids, because when you really start getting into it, there's not much difference between them. The transposable elements are bits of DNA that are sort of rogue. They act without the permission of the organism that they are living inside, and they can go back and forth. So a transposon can become a plasmid, which can become a virus. So there are some viruses that may have originally been transposons in other organisms, and some transposons may have once been viruses that integrated into the chromosome. So this is what you get in the blending of the living and non-living genetic worlds. This gray area, there's this whole world of genetic elements that are rogue, and they affect the lives of living things in profound ways. So if you're really interested in how transposons have changed evolution, you should look at, you can read about placenta, the evolution of placenta. So like I said, transposons and viruses go back and forth. A virus that inserts its DNA into the host cell chromosome. We'll talk about that a lot in the virus chapter. There's many viruses that they're called retroviruses, and bacteriophages can do this too. What they have to do is they inject their DNA and they actually insert it into the host cell's chromosome. Chickenpox is like that. Herpes viruses are like that. HIV is like that. They insert their DNA into the host cell chromosome and then they just hang out there. So now they're a part of the host's chromosome. They can never be removed. So that's, for a virus to insert itself into the DNA, it has to have the same genetic features as a transposon, which cuts itself out of the DNA and inserts itself elsewhere. Okay, so we know what that genetic code is. We can find viruses and transposons that have become part of our chromosomes because they have genetic features that encode for their capabilities of doing that, of cutting themselves out and reinserting themselves. So we can find these transposons in our genomes. We can find these retroviruses in our genomes, and it turns out that the genetic instructions for placenta are part of a transposon, which means it could have come from a virus, could have come from a different organism and inserted itself. So what viruses also have to do and this will all make sense, this is a blend of the viral chapter and the genetics chapter. So again, this is going to make a lot of sense when we learn about viruses because this is very common for viruses. So for viruses to get into the host cell, they often have to fuse. So they often have this lipid envelope. They have to latch on with proteins to the host cell and then fuse with it, empty their genes, and then maybe insert their genes into the host chromosome. Now, placentas do the exact same thing. When a mammal embryo is forming, the cells of that embryo fuse with the cells of the mother, and that's what creates the placenta. The genes for doing that are the same as viral genes, and they're preceded by these transposon genes. So there's a huge amount of evidence that the ability that basically we have incorporated into our genome this viral ability to fuse with a host cell, and that's what creates the placenta. A virus that many viruses can do that, and one of those viruses inserted itself in just the right place where that became part of our reproductive pattern. And so we are sort of a blend between reptiles lay eggs. They don't fuse with the mother. the mammalian ability for the infant to fuse cells with the mother's uterus, that is encoded by a virus that is now integrated into our genome. It's part of a transposon that's now become a part of our genome. So that's sort of, there's just these crazy impacts that these things can have on evolutionary trajectories. So would you expect, now most of the time transposons are going to be bad, right? Because would you expect transposons to be more or less dangerous than a base substitution mutation or than a frame shift mutation? So you've got this huge segment of DNA that just pops itself into the middle of the genome somewhere. Usually that's going to be massively disruptive, right? But just by chance, life has been on this planet for billions of years. Just by chance, sometimes it's not going to be totally disruptive. It might just create something new.